Hello and welcome to the presentation of my doctoral research at the KU Leuven. My name is Tianlu Wang and my PhD thesis is titled Closing the Loop Towards Contemporary Assessment and Modulation of Visual Attention. We live in a very rich sensory world. Natural visual scenes, such as this view of the Grote Markt in Leuven, contain many different cluttered bits of information. In fact, whenever our eyes are open, we receive an average of 1 billion bits of visual information every second. That's a lot of information. It far exceeds what our brain is capable of processing. However, we still manage to navigate through our day-to-day activities, and we seem to have a complete view of our surroundings without feeling completely overwhelmed. This is because of visual attention, the cognitive process that mediates the selection of relevant information and the filtering out of irrelevant information from cluttered visual scenes. It guides our visual processing and essentially controls what we become aware of in our environment. It goes without saying that visual attention is crucial to our everyday life. Unfortunately, this process can become impaired in many different ways, affecting how we determine which objects we attend, how we direct our attention to an object, and how to keep our attention at that location. Impairments in visual attention often occur as a result of injury to the cerebral cortex. This happens most commonly after stroke. A stroke occurs when a blood vessel that carries oxygen to the brain is either blocked or ruptured. When that happens, part of the brain cannot get the oxygen it needs and dies. Stroke is one of the leading causes of disability worldwide. Currently, the lifetime risk of stroke in the global population is 1 in 4 people over the age of 25. Of these stroke patients, about half experience attention deficits. If the damage occurs to the right parietal cortex, highlighted in this figure to the left, this percentage increases to over 80%. For most people, the symptoms generally disappear after a couple of months. However, they will remain for every 1 out of 4 or 5 patients. That is why it's very important to develop methods that could aid in the rehabilitation of visual attention deficits. The rehabilitation of such a complex cognitive function is in itself a very complex and large undertaking. In my PhD project, I looked at 3 of the key components required for successful rehabilitation. First, we need to be able to capture accurate cognitive profiles of the affected patients and obtain a reliable diagnosis of visual attention deficits. In other words, we need an accurate assessment of visual attention. Second, we need to understand the neural mechanisms underlying visual attention. Finally, we need an intervention to modulate visual attention, tailored to the individual patient. My PhD was built on these 3 components. In my thesis, I started with an investigation of accessible and inclusive assessments of visual attention. Then I continued to look at the neural mechanisms behind directing attention to locations across space. Finally, I investigated methods to self-modulate visual attention. First, the assessment of visual attention. Currently, the most commonly used clinical measures of visual attention are so-called paper and pencil tasks, where participants are asked to copy a figure, draw a clock from memory, or cross out all complete hearts on a piece of paper. These tests are simple, fast, and they perform relatively well in diagnosing prominent visual attention deficits. However, one drawback is that they often lack the sensitivity to diagnose the more subtle deficits in visual attention. Furthermore, the accuracy of their attention measures rely on the integrity of other functions, like motor functions and short-term memory. When these functions are damaged, as is often the case after a stroke, this would lead to attention measures that may not be accurate. In comparison to these paper and pencil tasks, computerized scientific paradigms from the fields of experimental psychology and cognitive neuroscience may bring many benefits to the clinics. In particular, these paradigms are specifically designed to provide accurate and sensitive measures of different components of attention. One such paradigm is based on the theory of visual attention, or TVA for short. TVA is a mathematical model that can provide quantitative measures of separable components of attention. The paradigm itself is a reporting task, where participants are presented with 2 or 6 letters in red or blue, and they're asked to report all the red letters that they can see. Using computational modeling on this behavioral data, we can derive several parameters of attention. These include measures of visual short-term memory capacity, perceptual threshold, processing capacity, efficiency of attentional selection, and Way index, which is the spatial bias of attention across the left and right visual field. In summary, these types of tasks based on the theory of visual attention are sensitive, accurate, and can provide measures of distinct components of attention. They're also especially suitable for stroke patients because they don't rely on intact motor functions. Instead of being based on reaction times, they're based on accuracy. However, one challenge in using such computerized tasks for clinical purposes is that they cannot be easily incorporated into clinical settings. This is because these lab-based experiments are usually performed with high-tech devices in controlled environments. Another potential challenge is the fact that these TVA-based tasks are usually performed using letter or number stimuli. This may make the task difficult to perform or even inaccessible to individuals who have difficulties processing alphanumeric stimuli. In the first part of my PhD, I investigated whether lightweight and portable devices, such as tablet PCs, can facilitate the translation of these computerized tasks into clinical settings, as they are generally more accessible. In addition, I investigated whether non-alphanumeric image stimuli can be used in order to make the task more inclusive for individuals with letter processing difficulties. To explore these possibilities, I compared the reliability of lab-based TVA assessment to that of tablet-based assessment, and the reliability of TVA assessments using letter stimuli to that of assessments using images as stimuli. The results indicated that both variations have a comparable reliability. In conclusion, TVA-based tasks performed on a tablet and using image stimuli are similar in reliability as the conventional lab-based TVA task using letter stimuli. They seem to be a promising and accessible tool to acquire clinical measures of visual attention at the bedside of the patient. However, more research is needed with larger groups of patients and in more environments resembling the real world. After the assessment of visual attention, the next step was to investigate the neural mechanisms behind visual attention. First, some background on how our attention works. Attention is distributed across the visual field according to both physical and behavioral attributes. Examples of physical attributes that tend to draw our attention are regions in the visual field with high contrast, such as edges or lines. An example of a behavioral attribute is when you're looking for someone who's wearing a red shirt. In this case, all red objects in the visual field will stand out. The brain then combines this information and produces a so-called Attentional Priority Map. This map guides our visual attention and determines how our attention is distributed across the visual field. Previous research has found that a brain region that plays a key role in the construction of the Attentional Priority Map is the intraparietal sulcus, or IPS for short. Now, this all seems pretty straightforward. However, there are a few more interesting findings in previous literature regarding the distribution of attention across the visual field. Namely, researchers have found that typically attention tends to be more distributed towards the left side of space, something referred to as leftward attention bias. Another common behavioral observation is the center bias. This means that attention tends to be distributed towards the center of the visual field, and perceptual performance decreases towards the periphery. Now, since the intraparietal sulcus is involved in the distribution of attention across the visual field, this made me wonder, how does attending to the left versus right visual field affect the neural activity within the intraparietal sulcus? Simultaneously, what are the neural effects in the intraparietal sulcus of attending to the central versus peripheral visual field, also referred to as at low or high eccentricity? This was the question in the second part of my PhD. I measured the brain activity of healthy participants while they were instructed to attend to a location in the left or right visual field, either at the center, which is also referred to as low eccentricity, or in the periphery, which is referred to as high eccentricity. At the same time, I also measured their perceptual performance on a reporting task. The results showed that participants performed better on a perceptual task when the stimuli were presented closer to the center at low eccentricity compared to the periphery at high eccentricity. However, their neural activity did not differ when attending to locations at low or high eccentricity. There were no differences between attending to the left or right visual fields on the behavioral level. On the other hand, we found a stronger activation in the left intraparietal sulcus following attending towards the right visual field compared to the right intraparietal sulcus. In conclusion, we found behavioral but no neural effects of the eccentricity of the location to which visual attention is directed. Based on this difference between the behavioral and neural effects, we suggest that future studies might investigate the eccentricity effect after increasing the top-down control of attention, for example, through increasing the task difficulty. Finally, now that we have a method to acquire sensitive measures of visual attention and the background knowledge of the neural mechanisms, we arrive at the last section of my PhD, the modulation of visual attention. Here, I want to briefly take a moment to talk about hemispatial neglect, a neuropsychological disorder quite common after stroke, in which visual attention is disrupted in the spatial component. Simply put, hemispatial neglect is a condition that causes people to be unable to pay attention to one side of space, usually the left side. They are unaware of stimulation from half of their world, as if that half just doesn't exist. The patients may even neglect their own body parts. In addition, a general reduction of alertness is commonly observed across the entire visual field, making hemispatial neglect a very multidimensional and heterogeneous syndrome. Current clinical methods to treat the symptoms of neglect include visual scanning and actively reminding patients to attend to the left, prism adaptation, which forcefully shifts their field of view to the right, thereby shifting parts of their left visual field into view, and a relatively new method called neurostimulation. Now, in particular, neurostimulation has shown promise in improving the spatial symptoms of hemispatial neglect by targeting the disrupted functions at the neural level. We already mentioned the intraparietal sulcus before. It's a region involved in the distribution of attention across the visual field. The spatial attention bias characteristic of hemispatial neglect has been related to an imbalance between the neural activity in the left and the right IPS. Recovering this activity imbalance has been shown to improve the asymmetrical distribution of attention. Although the effectiveness of neurostimulation seems promising, there is insufficient scientific evidence to support its transfer to daily life. And most importantly, these methods, which rely on external stimulation, are not suitable for a lot of patients. A non-invasive method to modulate brain activity without external stimulation could be a useful addition to the current clinical methods. One such method is through real-time fMRI neurofeedback. Neurofeedback is a technique that trains participants to control their own brain activity in a specific predetermined region by finding a personal mental strategy that works best for them. Participants receive information about their ongoing brain activity in real time, and they can use this information to adjust their personal strategies. When the participants learn to modulate their brain activity voluntarily, this would then lead to changes at the behavioral level. In the current study, we want to examine if one can use neurofeedback to voluntarily control the interhemispheric IPS activity balance in an endogenous way without external stimulation. We first conducted a systematic review of the literature and found that real-time fMRI neurofeedback has been successfully applied in both healthy individuals and stroke patients to modulate their own brain activity. It can also be used to treat motor deficits after stroke. The previous literature also showed some indications that neurofeedback can be applied to self-modulate cognitive functions, including attention. Next, we set up a proof-of-concept study to examine the feasibility of using real-time fMRI neurofeedback to self-modulate the interhemispheric IPS activity balance. We trained 6 participants to control their IPS activity balance either towards the left hemisphere or towards the right hemisphere. The study took place over the course of 5 weeks. The participants took part in 3 weekly neurofeedback sessions. Before and after the training, as well as after each training session, we estimated the distribution of attention across the visual field using the TVA-based task that was also used in the previous study. We also collected information about the current mood and motivation level of the participants, as well as the strategies that they used. Over the course of the training, 2 of the 3 participants in the left IPS group increased the activity in the left relative to the right IPS, while the participants in the right IPS group were not able to increase their differential IPS activity. The spatial distribution of attention did not change over the course of the experiment for any of the participants. In conclusion, this study indicates that it is possible for healthy individuals to voluntarily modulate the interhemispheric IPS activity balance using real-time fMRI neurofeedback. Based on the difference between the leftward and the rightward group, we suggest that self-modulation of the interhemispheric IPS activity balance might be direction-specific. Further research would be required to examine this, and to examine the effectiveness of neurofeedback in individualized training protocols for hemispatial neglect. Looking back, we found promising results for the use of computerized assessments of visual attention and the self-modulation of interhemispheric IPS activity balance with the help of real-time fMRI neurofeedback. The work in this thesis formed a contribution to the research towards individualized rehabilitation protocols for visual attention deficits, specifically in the components of assessment and modulation. However, we need to keep in mind that these findings were obtained under the best possible circumstances, in controlled environments, rather than in a very rich sensory environment resembling the real world. Looking forward, more research is needed towards individualized assessment and modulation of visual attention. First, the results in this thesis need to be replicated in a larger cohort of healthy participants. In particular, future research should aim towards a better understanding of the neural changes associated with the specific neurofeedback training protocols. Second, the potential of the TVA assessment and the neurofeedback training should be examined in stroke patients. Optimally, individual patterns of impairments, as described using the TVA assessment, should be considered in the design of the individualized neurofeedback training. Third, avenues for future research should involve testing the generalizability of the findings to different clinical populations and to different brain networks and cognitive domains. Finally, I want to highlight the importance of a stronger collaboration between engineers, neuropsychologists, neuroscientists, and clinicians in order to optimally integrate current technological advances and novel research findings into contemporary rehabilitation of visual attention. I want to end by acknowledging a large number of people that have made my PhD research possible, including but not limited to the names shown on the screen. In particular, I want to thank all the volunteers who have participated in my studies. Thank you for watching and have a great day.